السلام عليكم ورحمة الله تعالى وبركاته. I am Selma Al-Mahdi. Welcome to our lecture today, titled Citric Acid Cycle. Our objectives include identification of the key enzymes and intermediates of the citric acid cycle, to discuss the role of some vitamins in the cycle, and to describe how the cycle is integrated with the metabolism of carbohydrate, lipids, and proteins, and to explain the central role of the cycle in generating energy, particularly in the form of ATP, and to calculate how much ATP is produced by the cycle, and how the cycle is regulated, and how is it linked to the electron transport chain, and finally, the clinical relevance of some of the enzyme deficiencies and inhibitors of the citric acid cycle. Citric acid cycle is also known as tricarboxylic acid cycle or Krebs cycle, described by Sir Hans Adolf Krebs, a German biochemist and physician who received Nobel Prize in recognition for his effort in describing the cycle. The cycle is an anabolic pathway, meaning that it functions in both oxidative and synthetic processes in breakdown and in building up of biomolecules. Citric acid cycle is the final common pathway for aerobic oxidation of carbohydrates, lipids, and proteins. Glucose, fatty acids, and most amino acids are metabolized to a common precursor, that is acetyl-CoA, or intermediates of the citric acid cycle itself. So, can you see the cursor? Carbohydrates broken down to glucose or monosaccharides, proteins into amino acids, fats into fatty acids and glycerol. All of them can be converted to anabolic intermediate acetyl-CoA or the intermediates of the citric acid cycle itself. This is followed by generation of reducing equivalents by the cycle, which proceed to the electron transport chain to be coupled to positive phosphorylation of ADP to form ATP. So that is how our body gets energy from food. A comprehensive definition of the citric acid cycle will be that it is a series of mitochondrial reactions that lead to catabolism of acetyl residues and formation of reducing equivalents, which enter the respiratory chain where ATP is generated, thus capturing most of the energy of tissue fuels. This definition here stresses upon the fact that it is a series of reactions. The site of the citric acid cycle being the mitochondria, acetyl-CoA being the starting molecule, and the function or the roles of the cycle in production of reducing equivalents. It is linked to the respiratory chain, and the final outcome, which is capturing of energy of tissue fuels from the combined action of citric acid cycle and electron transport chain. Regarding the layout of the cycle, we said the starting molecule is acetyl-CoA. It is combined with oxaloacetate to form citrate. Citrate or citric acid is an acid that contains 3 carboxyl group, and from here the name tricarboxylic acid cycle comes. There is loss of 2 molecules of carbon dioxide with regeneration of oxaloacetate. So there is no net gain or loss in this cycle. We start with acetyl-CoA and oxaloacetate, and we reform oxaloacetate back again. And that is why oxaloacetate is considered to have a catalytic role. It acts as an enzyme, as it enters a reaction and gets out without being checked. These are the actual reactions of the cycle. Are they 8 or 10? Depends on how you count them. Okay, I have listed the reactions here for you. The bullets refers to the 10 reactions, and the number, as you can see, refers to the 8 reactions. The difference comes from how you count the reaction. If the reaction of formation of acetyl-CoA is considered, then you get 10 reactions. However, some will argue that formation of acetyl CoA is not part of the proper or the core reaction of citric acid cycle, so it may not be counted. The second thing is that the second reaction, the isomerization of with the formation of isocitrate is a two-step reaction. If you counted them two, then the total will be 10 reaction. If you count them as one reaction, then the total reaction will be 8. So 8 or 10, it's not a big difference if you know your intermediates and your reactions and how they proceed. So the first reaction of the citric acid cycle is condensation of acetyl CoA, which comes from metabolism of carbohydrates, lipids, and amino acids with oxaloacetate. Oxaloacetate contains 4 carbon atoms. Acetyl CoA contains 2 carbon atoms. Please pay attention to the 2 carbon atoms of acetyl CoA marked here with a dot and an asterisk, a star. Condensation of acetyl CoA with oxaloacetate is catalyzed by citrate synthase with the formation of an intermediate that will be hydrolyzed into citrate with release of CoA and thiol. This reaction is physiologically irreversible, so it will proceed only in the direction of formation of citrate. The second reaction is an isomerization reaction. As we said, it occurs in 2 steps, dehydration and rehydration, catalyzed by aconitase, whereby citrate is dehydrated to cis-aconitate, which is rehydrated to form isocitrate. Pay attention, the 2 carbon atoms that come from acetyl CoA are still present. This enzyme, aconitase, is inhibited by fluoroacetate. Fluoroacetate can combine with oxaloacetate to form fluoroacetyl CoA, which will inhibit aconitase, leading to accumulation of citrate, our substrate here. The third reaction is an oxidative decarboxylation reaction catalyzed by isocitrate dehydrogenase, which requires ions such as magnesium, and NAD, as you can see here, is the reducing liquid. It will receive the hydrogen ion. So isocitrate will be oxidatively decarboxylated to form alpha ketoglutarate with loss of the ferrous carbon dioxide molecule. The hydrogen that is removed from isocitrate will be received by nicotinic dinucleotide to form NADH plus H. Pay attention to the 2 molecules of acetyl CoA are still present. That is to say, carbon dioxide is not lost from the actual or our starting acetyl CoA molecule. Again, this reaction is an irreversible reaction. The fourth reaction, is oxidative decarboxylation catalyzed by another dehydrogenase, which is alpha ketoglutarate dehydrogenase. It's a big complex enzyme which utilizes or make use of various vitamins and cofactors, including thiamine diphosphate, like CoA, FAD, CoA, and NAD. Again, here will receive the hydrogen and become a reducing glucose that will proceed to the electron transport chain. This NAD, of course, will be linked to complex 1. This irreversible reaction will lead to the formation of succinyl CoA. This reaction is inhibited by arsenate. The fifth reaction is what we call substrate level phosphorylation, catalyzed by succinate thiokinase or succinyl CoA synthase. So, is this a reversible reaction? You can name it succinyl CoA synthetase or succinate thiokinase, depending on the origin of the reaction. And this is substrate level phosphorylation, as you can see here. Guanine dinucleotide phosphate is phosphorylated to guanine trinucleotide, guanine nucleotide triphosphate with two high energy phosphates. The sixth reaction is oxidation of succinate, to form fumarate, succinate dehydrogenase in an irreversible reaction whereby FAD is reduced to FADH2, and this is linked directly to Q component of ubiquinone component of the electron transport chain. This reaction can be inhibited by malonate, leading to accumulation of succinate. The second reaction is a hydration reaction whereby fumarase or hydratase adds water to fumarate to form malate. It is a reversible reaction. The eighth and last reaction is oxidation by dehydrogenation carried out by malate dehydrogenase, whereby NAD is reduced to NH2 to convert malate to oxaloacetate. NAD here receives the hydrogen to be reduced to NADH plus H plus, and of course will proceed to the electron transport chain through complex 1. This reaction here favors actually the formation of malate. However, if you want for the citric acid cycle to be complete, to regenerate oxaloacetate, you need to proceed from malate to oxaloacetate in this direction. So how to ensure that this reversible reaction goes only in the direction from malate to oxaloacetate? By continuous removal of the products of oxaloacetate. Oxaloacetate can proceed to, for example, transamination or gluconeogenesis, and NADH will proceed with electron transport chain or any oxidation reduction reaction. So by removal of products, you ensure that this reaction proceeds to completion, and by this we completed all the reactions of citric acid cycle. The net reaction is that you start with one molecule of acetyl-CoA plus 3 NAD plus 1 molecule of FAD plus 1 molecule of DDP, inorganic phosphate, and water. We removed 2 molecules of carbon dioxide in 2 separate positive decarboxylation, and we gained 3 NADH, 1 molecule of ADH2, 1 molecule of DDP, 2 hydrogen, and 4 electrons. Vitamins play a significant role in citric acid cycle. Namely, we have 4 water-soluble vitamins, and that is riboflavin as FAD, a cofactor for succinate dehydrogenase, niacin as an electron acceptor for the dehydrogenases for isocitrate, alpha-ketyl butyrate, and malate, thiamine as thymine diphosphate in the reaction that is catalyzed by alpha-ketyl dehydrogenase, and pantothenic acid defined as a CoA, part of the acetyl-CoA acid-CoA. So what is the energy yield of citric acid cycle? We said in the definition of citric acid cycle, we use it to harness or to capture the energy from carbohydrates, lipids, and proteins. For each cycle, dehydrogenases, as we have already seen, catalyze production of 3 NADH and 1 FADH2. This reducing equivalence, of course, will be transferred to the electron transport chain for the oxidation, whereby every NADH will give us approximately 3 ATPs. Every FADH2 will approximately give us 2 ATPs. And this is just to remind us again of the sites of production of reducing equivalence. NADH here, the first one, at the level of isocitrate dehydrogenase, the second NADH at the level of alpha-ketoglutarate dehydrogenase, the third NADH here at the level of malate dehydrogenase. FADH here is produced by succinate dehydrogenase, and we have here the only example of substrate level phosphorylation whereby GTP is produced. GTP of course is equivalent to 80. So if you want to calculate it as a net number of ATP molecules, every NADH is equivalent to 3 ATPs. Every ADH2 is equivalent to 2 ATPs. So 3 multiplied by 3 is 9 ATPs. 1 multiplied by 2 is 2 ATP. And then 1 GTP from substrate level phosphorylation, that is equivalent to ATP. When you sum them up, 9 plus 2 plus 1 equal 12 ATPs. So 12 ATPs are formed per one term of citric acid cycle. There one acetyl-CoA oxidized. How citric acid cycle is regulated? Citric acid cycle is tightly regulated to meet the cell's energy demands and at the same time to maintain the metabolic balance of the cell and of the body as a whole. Regulation occurs at several points actually, at the point of entry into citric acid cycle and the rate of the citric acid cycle itself. Control of entry is either by allosteric regulation, covalent modification, or hormonal regulation of the pyruvate dehydrogenase complex. Allosteric regulation includes inhibitors such as ATP, acetyl-CoA, and NADH, and activators such as ADP, CoA, and NADP. Note that ATP, acetyl-CoA, and NADH all signify abundant energy. When you have energy, you don't want to operate the citric acid cycle anymore, so you would like to inhibit it. On the other hand, ADP, CoA, and NAD signify low energy, so you want to operate your citric acid cycle. That is why ADP, CoA, and NAD activate citric acid cycle to operate it to provide energy for the body. Covalent modification of pyruvate dehydrogenase. Pyruvate dehydrogenase is inhibited by reversible phosphorylation by a specific protein kinase. So the phosphorylated form of pyruvate dehydrogenase is actually inactive. And pyruvate dehydrogenase is activated by dephosphorylation by a specific phosphate protein phosphatase. So the active form actually of pyruvate dehydrogenase is a dephosphorylated form. The third form of control of pyruvate dehydrogenase at the entry of citric acid cycle is by hormones. Vasopressin and alpha-adrenergic agonists increase intracellular calcium ion level, and this stimulates the phosphatases. That is to say, a phosphatase, of course, will dephosphorylate protein pyruvate dehydrogenase and lead to activation of the enzyme. Insulin also stimulates the phosphorylation of the complex. In this figure here, you can see this is the pyruvate dehydrogenase, okay, in the active form dephosphorylated, and this is pyruvate dehydrogenase in the inactive form phosphorylated. It contains a phosphate group. To interchange between the active and inactive form, you use a phosphatase and a kinase. A kinase will add a phosphate group to the active form, so it will be inactive. A phosphatase will remove the phosphate group from the inactive form to activate the enzyme. ADP, CoA, and NADP, as we said, are allosteric activators of the pyruvate dehydrogenase. They signify low energy, and they will lead to activation of the pyruvate dehydrogenase, which will lead to formation of acetyl-CoA, and acetyl-CoA will go into the citric acid cycle. While NADH here, NADH is a negative regulator because it signifies abundance of energy. That's to say, to stop the citric acid cycle from the acetyl. Acetyl-CoA is a product itself can negatively can form negative feedback loop to inhibit the pyruvate dehydrogenase complex. Hormonal regulation, insulin here. Insulin activates the phosphatase, and the phosphatase, of course, converts the inactive form to the active form. So we have three levels of regulation of pyruvate dehydrogenase. Regarding the regulation of the rate of the citric acid cycle, it is precisely adjusted to the cell's needs for ATP or for energy. It is reduced when the energy is abundant and vice versa. The rate of flux through the cycle is to say how does the cycle proceeds or turn on or get continuous progression is at three highly exogenic steps: at the level of the citrate synthase, at the level of the isocitrate dehydrogenase, and at the level of alpha-ketoglutarate dehydrogenase. Controlling these enzymes will control the rate of the cycle. Citrate synthase can be affected by the availability of the substrate, the acetyl-CoA and the acetate. If they are limited or low, then we expect that the rate will be low and vice versa. Citrate synthase can be inhibited by accumulating citrate and succinyl-CoA. Citrate, of course, is the immediate product for this reaction, so it negatively inhibits the reaction. ATP is an inhibitor because ATP means that there is a lot of energy and you need to stop the cycle, while ADP will be an activator. Why? Because ADP means there is low energy or no energy and the cycle needs to be activated. Isocitrate dehydrogenase also is allosterically regulated, inhibited by NADH and ATP. If there is energy, you need to stop the cycle to inhibit the cycle. If there is NAD, ADP, magnesium or calcium, you need energy, and that's why you need to activate the cycle. Alpha-ketoglutarate dehydrogenase is allosterically inhibited by succinyl-CoA and NADH and activated by calcium. I would like you here to pay attention to how the regulation is coordinated along the pathway. If, for instance, here you have ATP negatively inhibiting pyruvate, ATP here negatively inhibiting citrate synthase, ATP here negatively inhibiting isocitrate dehydrogenase, and AMP at the same time can activate the pyruvate dehydrogenase. ADP can activate citrate synthase, and ADP here can activate isocitrate dehydrogenase. So when there is a lot of energy in the form of ATP, you need to stop the cycle. So you will stop pyruvate dehydrogenase, you will stop citrate synthase, and you will stop isocitrate dehydrogenase. And when the energy is low, signified by the AMP or ADP, you need to phosphorylate these residues to form ATP. So you need to operate the cycle. That is why you need to activate pyruvate dehydrogenase, activate citrate synthase, and activate isocitrate dehydrogenase to operate the cycle to produce reducing equivalents to proceed into the electron transport chain to produce ATP. So whenever there is energy signified by different molecules and substrate, you need to stop the cycle completely. When there is low energy signified by substrates and molecules such as NAD, ADP, AMP, CoA, then you would need to activate the cycle to produce energy. They are contradictory situations. Citric acid cycle plays major roles in metabolism in general, so it is a final common pathway as we have seen for the oxidation of metabolic fuels. It participates in gluconeogenesis, which is synthesis of glucose from non-carbohydrate pyruvate intermediates. It participates in transamination and deamination, which are reactions that are we will discuss them as we come to protein metabolism, and it participates in fatty acid synthesis. Being the common pathway has been already discussed. The role of citric acid cycle regarding gluconeogenesis: all the intermediates of the citric acid cycle are potentially glucogenic. So any intermediate of the citric acid cycle, we can use it to form glucose in a process that is called gluconeogenesis. We have a net transfer out of the cycle through the action of phosphoenol pyruvate carboxykinase, which decarboxylates oxaloacetate to form phosphoenol pyruvate. Phosphoenol pyruvate is a shared intermediate between glycolysis and gluconeogenesis. We have net transfer into the cycle by two enzymes, a pyruvate carboxylase and a lactate dehydrogenase. Let us discuss this further. We want to understand how citric acid cycle is being part of glycolysis or of carbohydrate metabolism. Glycolysis is a process of oxidation of glucose. It can either end up in lactate or pyruvate. So lactate and pyruvate are both end products of glycolysis, of complete oxidation of glucose. If you want to proceed further, so the body has oxidized glucose in order to gain energy, but energy is still harnessed here in lactate and in pyruvate. They have to go through the citric acid cycle to form reducing equivalents, to go into the electron transport chain to catch them as ATP. That is how the body produces energy from carbohydrate. So how does the end products of glycolysis gain entry into citric acid cycle? Through the enzyme lactate dehydrogenase and pyruvate carboxylase. Lactate dehydrogenase convert lactate to pyruvate, and pyruvate then is converted by pyruvate carboxylase to oxaloacetate, and now oxaloacetate can proceed through the citric acid cycle. Again, you started with glucose that has been oxidized to either lactate or pyruvate. If it has been oxidized to lactate, lactate has to be dehydrogenated to pyruvate and then carboxylated to oxaloacetate, and it goes into citric acid cycle to produce reducing equivalents, and then the electron transport chain to produce ATP. When you link respiration, of course, with oxidative phosphorylation through the ATP synthesis. Glycolysis can alternatively in pyruvate, which through one step of carboxylation will form oxaloacetate. So how do we gain you get entry? We have already discussed entry, how to get out of the cycle. As we said, all the intermediates of citric acid cycle can be glucogenic, so you can use them to produce glucose. How is that? This will be through oxaloacetate. So if you want to form glucose from alpha ketoglutarate, it will have to be converted to succinyl CoA and to fumarate oxaloacetate. If you want to use fumarate as a glucogenic intermediate to produce glucose, you have to convert it first to oxaloacetate. So once your intermediate is converted to oxaloacetate then under the action of phosphoenol pyruvate carboxykinase will form phosphoenol pyruvate. Phosphoenol pyruvate, or PEP, is actually an intermediate in glycolysis and gluconeogenesis as well. So once phosphoenol pyruvate is formed, then that means you are within the pathway of glycolysis. So glycolysis is connected by net entry. If you want to take the products of glycolysis into citric acid cycle, or if you want to use the products or intermediates of citric acid cycle to go into glycolysis. Citric acid cycle is related to protein metabolism because citric acid cycle intermediates such as oxaloacetate and alpha ketoglutarate are the carbon skeletons are the same as for amino acids. Transaminases catalyze transamination reaction which are reversible interconversion of amino acids and their alpha keto acids analogs. For example, the carbon skeleton of alanine, that is, it is carbon skeleton minus or without the amino group, is actually pyruvate. When you add the amino group to pyruvate, you get alanine. When you remove the amino group from alanine, you get pyruvate. This is a reversible reaction. Both of them are C carbon atom. Likewise, the carbon skeleton, the 4-carbon atom skeleton of aspartate, is actual oxaloacetate plus an amino group. And that of glutamate is actually alpha glutarate plus amino group. When you remove the amino group from glutamate, you get alpha glutarate, and when you add it, you get glutamate back. But the transaminases catalyze the interconversion of the amino acids with their carbon skeleton. So what is the relationship between citric acid cycle and amino acid metabolism and protein metabolism? For instance, we can use intermediates of citric acid cycle, oxaloacetate, for example, to form aspartate. We can use alpha ketoglutarate to form glutamate, and this connects citric acid cycle with amino acid metabolism. On the other hand, we can use certain amino acids to produce intermediates of citric acid cycle. As the stars here refers to different amino acid. We will talk about them when we come to protein metabolism lectures. For example, 2 amino acids here can be degraded to fumarate. 3 amino acids can be degraded to succinyl-CoA. 4 amino acids can give you glutamate, and then from glutamate you go to alpha ketoglutarate. So the connection between citric acid cycle and protein or amino acid metabolism is that some intermediates of the citric acid cycle can form amino acids, and some amino acids can be converted to intermediates of the citric acid cycle. What is the role of citric acid cycle in fatty acid synthesis? In non-ruminants, including humans, acetyl-CoA is the main substrate for fatty acid synthesis. So if you want to build up a fatty acid side chain in your body, you will start by acetyl-CoA and add it sequentially until you complete your fatty acid molecule. Acetyl-CoA is formed from pyruvate by the action of pyruvate dehydrogenase. We have a dilemma here. Pyruvate dehydrogenase is a mitochondrial enzyme. It is needed, as we said, for formation of acetyl-CoA from pyruvate. Acetyl-CoA is the building block for fatty acid. Fatty acid synthesis occurs in the cytosol, and the mitochondrial membrane is impermeable to acetyl-CoA. So if you use pyruvate dehydrogenase to convert pyruvate to acetyl-CoA, your building block, you will use acetyl-CoA to build up your fatty acid. However, this step, the formation of acetyl-CoA, occurs in the mitochondria, and the pathway for fatty acid synthesis occurs in the cytosol. The two processes, the formation of the first substrate, acetyl-CoA, is in the mitochondria. The proper pathway for fatty acid synthesis is a cytosolic process, 2 different compartments. How does the cell manage to move acetyl-CoA from within the mitochondria to the cytosol? Is by converting acetyl-CoA to citrate in the mitochondria through the first reaction of citric acid cycle. Citrate can cross a mitochondrial membrane easily, unlike acetyl-CoA, and in the cytosol will be reconverted back to acetyl-CoA by citrate ligase. Transport of citrate outside the mitochondria occurs after saturation of aromatase to ensure that citric acid cycle, that fatty acid synthesis occurs only if the citric acid cycle is saturated. Let us recap this again. We said acetyl-CoA is used as the building block for fatty acid synthesis. However, acetyl-CoA is produced within the mitochondria. This is the mitochondrial membrane. Acetyl-CoA is produced inside the mitochondria by mitochondrial enzyme pyruvate dehydrogenase from pyruvate. Pyruvate comes from the cytosol and crosses as a mitochondrial membrane to be dehydrogenated to acetyl-CoA. If you want to proceed to fatty acid synthesis, you need to transport acetyl-CoA from inside the mitochondria to the cytosol. Acetyl-CoA cannot cross the mitochondrial membrane, so the cell manages time converting acetyl-CoA to citrate by citrate synthesis. Citrate easily cross the mitochondrial membrane. In the cytosol, citrate will be reconverted back to acetyl-CoA by citrate lyase, and acetyl-CoA then will proceed to fatty acid synthesis. So this means there is competition for acetyl-CoA between fatty acid synthesis and it is pathway in the citric acid cycle. Whether this acetyl-CoA to be committed for the citric acid cycle pathway or to go outside for fatty acid synthesis. They are two different pathways. Citric acid cycle here represents degradation pathway for energy production, while fatty acid synthesis is an anabolic pathway for storing energy as fatty acid. The priority here will be for citric acid cycle. So as long as citric acid cycle needs to go on, all the acetyl-CoA will proceed through the reactions of citric acid cycle. If the citric acid cycle, then if there is enough energy, then the surplus acetyl-CoA will be transported to the cytosol as citrate to be converted to fatty acid. So this makes sense in the sense that as long as there is energy needs, there is need for ATP, also acetyl-CoA will use in citric acid cycle to eventually produce ATP. Once there is no longer need for ATP production or the need is low, some of the acetyl-CoA can go outside. This surplus acetyl-CoA will be stored as fatty acids. There are very few reported genetic abnormalities of the enzymes of the citric acid cycle, meaning that they are incompatible with normal development or with life. When there is a deficiency of citric acid enzymes, you expect neurological damage due to impaired ATP and energy metabolism in the central nervous system and in the neurons. You expect also involvement of the liver in form of hepatitis and cirrhosis. Why? Because the liver has a central role in metabolism and pathways such as gluconeogenesis, lipogenesis, and many pathways will be inhibited with accumulation of substrates and toxicity to the hepatocytes. You also expect muscle weakness and lactic acidosis because muscles no longer find the energy from the citric acid cycle and electron transport chain. This is the list here of the enzymes and their associated manifestations. That is all. Thank you very much.